All right, this is part 2 of chapter 7. We're starting on slide 35 with the blood collection additives. All right, so the type of additive required for each tube that you draw, generally it depends on the test that's ordered. Some tests that are ordered can technically be done on different kinds of tubes. So for instance, a lipid panel. Okay, that could technically be done on probably a gold top, a red top, or a green top. But it's going to depend on the facility and what kind of instruments that they have that run the test. What kind of equipment are they using? How are they doing this test? What reagents are they using? So that's going to dictate exactly which tube your facility might have you collect a lipid panel in a gold top, whereas your classmate's facility might have it collected in a green top or a red top. It just depends. So sometimes I'll be able to tell you this test always goes in this tube. Sometimes I will not. It just sort of depends on the type of testing, and hopefully you'll get to understand how I figure that out as you go on. We'll talk about that a lot in lab as we go. Additives come in different forms, so they'll be liquid, they could be spray dried in the inside, or powder. Sometimes you look inside a tube, you'll be able to see something inside of it, and sometimes you won't, okay? All additive tubes, basically every tube that we're going to draw for testing has an additive of some kind in it, and it has to be inverted. If you read in your chapter, each section on each of these tubes, additives, will tell you exactly how many inversions each additive requires. Some inversions only, or some additives only require 3 inversions, some require 10. If you do 8 to 10 inversions for every single tube, you'll be safe. You can't over-invert, but you can under-invert it. Basically, inverting the tube mixes the blood inside the tube with the additive that's inside that tube, so that's evenly distributed, and either prevents the blood from clotting, all of the blood from clotting, or it makes it so that all the blood clots at a nice even pace. And 1 inversion equals taking the tube upright, turning your wrist 180 degrees so the tube goes all the way upside down and then all the way back up again. That's 1 inversion. And we will demonstrate what that is in class. Oops, too far. Anticoagulants are one of the additives that you're going to see in your tubes, and they prevent blood from clotting or coagulating. Okay? It keeps it in the liquid form. It keeps it in whole blood form. Okay, and it does it through a number of ways. It either does it by chelating or binding or precipitating calcium. So basically it takes that calcium and it attaches to it or puts it out of the specimen in such a way that it makes it unavailable to the coagulation process. Your blood has a natural process that it goes through to clot, and it needs calcium to do that. It also needs thrombin to do that. And naturally, the blood would start to form thrombin through a certain number of chemical reactions. We'll talk about that in chapter 6. So other anticoagulants work by inhibiting that formation of thrombin so that it can't do the clotting process through fibrinogen to fibrin and all this other stuff. Okay? So if a test requires whole blood meaning what it looks like inside your body, not clotted, whole blood, or it needs plasma, which would be whole blood that's been spun down in a centrifuge and had the red blood cells and the white blood cells pulled out and just the liquid portion, it must be collected in a tube with an anticoagulant. Okay, so that's these tubes that we're going to talk about. On page 199, I think those page numbers are correct. There should be a little memory jogger about the anticoagulants. I think it's something like ECHO. You have to prevent microclots from forming, so that means you have to invert your tubes right away. So here are your most common anticoagulants. We've got EDTA, which are your purple and pink ones on the right side of this picture here. You have citrates, which would be the blue top, light blue top. Okay, we also have another really tall, skinny black tube that has sodium citrate in it, but we don't use it very much, but we'll talk about that in lab. We have heparin tubes, which are the green tops, whether it's mint green, dark green, green with like a tigery top kind of thing. It doesn't matter as long as it's green. And then you have oxalates, which are gray, which I don't have in this picture here. Okay, so EDTA, they come usually powdered, and it's usually or sprayed inside. You can't usually see it. It chelates calcium, so it takes the calcium out of the picture. All right, you're going to see this in lavender tops, pink tops, and pearl top tubes, as well as the royal blue top tubes with the lavender label, although the ones in lab are kind of a dark blue more than they're lavender. Okay, you'll see it in the micro collection tubes that are lavender colored or pink kind of colored. Okay, you'll see it used in blood bank sometimes. Used to be they used red tops in blood bank, but now more and more places are switching over to the pink tops for that. Okay, you'll always see purple tops or lavender tops or whatever you want to call these kind of tubes in hematology. Okay, it preserves the cell shape and structure. It inhibits platelet aggregation better than other anticoagulants. And when we're in hematology, we're looking at the cells themselves, the red blood cells, the white blood cells, the platelets. We want to be able to see those structures clearly. So we want to make sure we use an additive that really clearly preserves our specimen and prevents it from clotting. All right, our next one is citrate, sodium citrate. So here's that skinny black tube. We use that for ESR, but we also, generally, we're not going to talk about that right now. We're just going to stick to the basics. So sodium citrate are these light blue top tubes. They prevent coagulation by, again, chelating calcium. The most common is sodium citrate. You're going to see those most often in the coagulation department of the lab, and it's going to work. Most common test is probably a PT or a PTT. It's a prothrombin time. If you know anybody on blood thinners and they have to have that tested regularly, they get a blue top tube drawn to test for that, okay? Excuse me. The reason that they like these in the coagulation department is because sodium citrate does a really good job of preserving the particular coagulation factors, which we haven't talked about. That's in chapter 6. And that's really, really important for when we're trying to test for those specific factors and how well do you have a clotting disease, then we want to look at those factors. So again, we have to immediately mix them. You're going to see that on every one of these, okay? Coagulation tests are performed on plasma. So that means that we have whole blood goes into this tube, and then the tube gets spun down, and then that separates the cells, the red blood cells, white blood cells, and platelets from the liquid portion of the blood, and we're left with plasma. Okay, so that's what actually gets tested. Unlike hematology, where the whole blood is tested. Okay, they put it on a slide, they look at it under scope, they don't spin it. All right. Blue top tubes must be all the way full to maintain a 9-to-1 ratio of blood to anticoagulant, or the specimen will be rejected. Remember that this is really important when we're using butterfly needles with that tubing. We want to make sure we use a discard tube so that we don't get the air in our tube, so that we have a full tube, all right? Heparin tubes prevent thrombin from forming, okay, and that's how it prevents clotting from happening. You'll see heparin tubes used often in chemistry tests, especially a stat test, because it doesn't have to, we're going to talk about that later. It doesn't have to wait to clot to be tested. It can just be thrown right in because it's got an anticoagulant in it. You will also see that in your green top tubes, your light green, dark green, royal blue top tubes with a green label on it, and the micro tubes with the green writing or a green lid on them. Heparin comes in 3 flavors: ammonium heparin, lithium heparin, and sodium heparin. Lithium heparin is the most common one that's used. It generally comes with a gel on the bottom of the tube, but you will see the other ones used as well. Ooh, it went too fast. Oxalates are going to precipitate calcium, meaning it's going to take that calcium out of there and make it so you can't use it, so it prevents the tubes from clotting. Potassium oxalate is the most often used anticoagulant of the oxalates. It's usually added to tubes with a glucose preservative which is an antiglycolytic. Usually you'll see that in the form of sodium fluoride, but you can see them individually. You can see a gray tube with just sodium fluoride, which means it doesn't have an anticoagulant, which means it will clot, but it does preserve the glucose. You can also see the potassium oxalate all by itself without the glucose preservative. So again, you just have to check your tubes. All right, those are going to be your gray tops. Some special anticoagulants, you're going to see ACD, acid citrate dextrose. You're going to see ACD-A and ACD-B tubes. You use those in immunohematology or blood bank testing for like paternity evaluation, phenotyping, transplant compatibility. It binds to calcium, so it prevents it from clotting. The dextrose in it is actually a red blood cell nutrient, so it helps keep the red blood cells alive. And these are usually glass top tubes as well. They have a yellow top. CPD is citrate phosphate dextrose. That's usually used in the bags that we donate our blood in, right? The citrate keeps it from clotting by chelating the calcium. The phosphate stabilizes the pH, and the dextrose provides the red blood cells with food and keeps them alive, because we want our red blood cells to be alive when they get transfused into us, right? SPS tubes, or sodium polyethyl sulfonate, is used for blood culture collection. It's this large yellow top tube. Looks a lot like an ACD tube, but it's a little bit bigger, and it says sterile on the outside. It also prevents calcium, and this is an in-between tube between doing a blood culture test. We'll talk more about that when we talk about blood cultures. Antiglycolytic agent, again, that prevents glycolysis. You're going to see the most common one is sodium fluoride. I talked about that already, so you can read this on your own. All right, so some other tube additives are clot activators. These enhance coagulation. They activate the clotting process, right? It's basically going to add a substance into the blood that's going to include more surfaces for platelets to activate on. So you're going to have little silica particles or clays or more thrombin, okay? The most common one we're going to see is in our SST tubes, the silica, okay? Added to SST tubes, those are your red and black tubes, or your gold top tubes. That's an SST, a serum separator tube. That gel on the bottom is what signifies that it's a serum separator tube. You're also going to see that silica added to the plastic red top tubes. It causes the blood to clot in about 15 to 30 minutes rather than what would naturally occur in about 30 to 45 minutes. All right? You still have to mix your tubes or the particles get suspended and it doesn't work right. This gel on the bottom is called thixotropic gel. It doesn't actually interact with the specimen. It's usually located in the bottom of the tube. And the density is somewhere between the cells and the liquid portion of blood. So when you centrifuge it, when you spin it down, the gel becomes lighter than the red blood cells and it moves up to the center and it separates the red blood cells, which will be at the bottom, from the liquid portion, which will be at the top. Okay? Royal blue top tubes are trace element free tubes. They're made out of a special plastic that has as little of these trace elements as possible. So copper, lead, and zinc, they're naturally occurring in a lot of plastics. And they leach into our specimens. And if we were using a regular purple top or green top or a red top, and we were testing for copper or lead or zinc, some of these things that are in such small quantities, selenium, they're in our bodies in really, really tiny quantities, and they're also in the plastics in really, really tiny quantities. So we don't want that to be in those tubes. We use a special plastic that doesn't have these elements in them, and we do these testing in that. We don't make all of our tubes out of that because it'd just be too expensive and not necessary. So you have royal blue top, which are these trace element tubes, and they basically are the same exact thing as a red top, a purple top, or a green top, except they're made out of this special plastic instead. So they have the royal blue top stopper, but then they have these labels that have a red, a green, or a purple label on them. Okay, and then you'll have the same thing. So EDTA, heparin, they align with those labels, and those would be anticoagulants, whereas the red label is got either no additive or serum, clot activator, and that would make it clot, okay? According to CLSI, you're supposed to draw them separately, but some facilities will have you draw them right in line with, you know, the lavender label with the lavender tubes, okay? So you just follow your facility's rules on that. All right, order of draw. The order of draw refers to the order we are supposed to draw the tubes as far as their color is concerned. So, or their additive, really, it's the additive. So the order of draw is determined by the additive and which additives can interfere with other additives, interfere with the testing due to cross-contamination by the carryover of these additives. And that carryover happens when we put the tube onto, say, an ETS, and we push that little needle sheath up, and we stick our tube on, and the blood flows in, and it mixes with the additive, and it could potentially touch that needle, and that additive could get stuck on that needle, and then when we take that tube off, put the next tube on, a little bit of that additive goes into the next tube, right, with the needle. It's theoretical. It may not happen every time, but it could happen. So we need to make sure that we follow this guideline set by the CLSI of what tubes to draw in what order so that we minimize the effects of that additive carryover. So the very first tube should be your sterile tubes, your blood cultures. It doesn't happen on every draw or very often, so we don't always have to worry about that. So generally we're starting with the light blue tops, our serum tubes, which can be our red tops, our gold tops, with or without gel, it doesn't matter. As long if it gives us a clot activator in it, it goes next. Then all of our heparin tubes, with or without gel, it doesn't matter. Our EDTA tubes, lavender or pink, with the gel or not. And then our glycolytic inhibitor tubes, which are gray top tubes, okay? Now, when we work with finger sticks, capillaries, we're going to have a slightly different order of draw, and we'll talk about that when we get to that chapter. If you're in the middle of the draw and you realize that you need a tube that you should have drawn earlier, draw an extra of that same tube, use that first one as a clear tube or discard tube, and then collect your tube. So for instance, say you're drawing all your tubes, you get to the lavender and you realize, oh my goodness, I forgot to draw a green top. Well, green top's supposed to come before lavender. So what you could do is take 2 green tops after your lavender, since when you realized it, right? So then you start drawing a green top, get a few drops of blood into that green top, take that tube off, toss it aside, it's your discard tube, and then fill your actual green top. And that's going to clear that needle of any of that additive that might carry over into your tube, okay? So you have a table in your book that tells you a little bit more about that. So this is that carryover cross-contamination. This can occur when the blood in the additive tube touches the needle during ETS, when you transfer blood using a transfer device, using a syringe. EDTA is the one that's going to cause the biggest amount of problems. All right. Trace element tubes are also going to be more susceptible. Heparin causes the least amount of trouble because it occurs naturally in our blood. Tissue thromboplastin, this is something that can contaminate our specimens. It's present in tissue fluid. It happens basically as soon as you puncture the skin with, say, a needle or a lancet. It basically is a little chemical signal that activates one of the pathways in the clotting process and can interfere with some of our coagulation testing. So coagulation testing goes in light blue top tubes or sodium citrate tubes, and it doesn't bother our regular tests like PTs and PTTs. So we don't have to worry about that when that is an issue. But when we're doing coagulation testing of like factor 5 or factor 3, or there's a million of them, you don't have to look, you can go look them up on your own, but I'm not going to talk about them. We have to use a clear tube. It's basically to counteract the effect that this tissue thromboplastin chemical can have. We don't want that getting into our specimens for some of these other coagulation tests, or it can throw off the results. So we use a clear tube or a discard tube to collect that blood that has that tissue thromboplastin in it. Okay, we also use it for butterfly collections on the first tube as well. Oops. Blood cultures detect microbes in the blood, and we have to do a special cleansing using a special cleaner and a special swab little scrubby thing. We will talk more about this in another chapter. But basically, the most important part of blood culture collection is the skin cleansing. Oh my goodness, all my things are going off. Hopefully you can't hear those. There's a case study here. We can talk about this in class, but I will talk a little bit about it here. Actually, no, we'll talk about this in class. So you have this here. There are some case study questions, and then the answers, and then there's some resources here. You can go click on all these links and see some more information as you see fit. All right, that's it for the second half of the Chapter 7 lecture. Again, you'll be able to find these under the Unit 1 videos to watch. I'm either going to put them there, or I might just list them right here on the Unit 1 resources, so I haven't decided yet. Anyway, if you have any questions, feel free to email me or ask me in class.